All right, and we are live with White Flame. He is here to talk about his research. He just got accepted as his PhD just got accepted. So congratulations. I know you came a long way, and I know it was a lot of hard work. So feel free to tell us about your research.
Thank you. So it's definitely been a long road. So my research actually didn't start off in the way that it ended. I started by working on uh, it was sort of a cross collaborative project between several different labs on campus. The idea was we were developing a new kind of device that would have made it possible to detect foodborne pathogens on a variety of surfaces and plant types. It was actually something we spun off into a company later, which unfortunately is no longer running as a company, but still it was a good experience as a whole. After a while of doing that, uh, it didn't. The problem with that kind of project is that it's very yes or no. Things either work or they don't, and usually that's not a good PhD. You want a PhD that's going to, if it lands on no, you're going to be able to continue doing stuff. So in that case, I worked with a couple of other labs to figure this out, and the idea was I worked on something called the satellite RNA, which you can kind of imagine as a virus of a virus. A plant virus gets into a plant, it starts replicating, it starts producing lots of protein, and then the satellite gets in. It's much smaller, it doesn't look like the virus, but it's able to steal viral components away for its own replication and packaging. And so I was mainly interested in how it did that. That was the focus of my PhD.
Okay, sure. So is a satellite virus basically a virus that infects a virus?
So it could be seen that way, but not quite. So there are actual couple of different things that are kind of similar in that regard. So if you want a virus that infects a virus, there's actually things called virophages, which are kind of like bacteriophages except for viruses. And bacteriophages infect bacteria, virophages infect viruses, but they're these really tiny viruses that infect these gigantic viruses that infect amoeba mainly. That's a specific group. There's also satellite viruses, which are um, they still take things from a virus, but they produce their own coprotein, so they package themselves. They just require the virus for everything else. It's sort of like it's parasitizing the virus. It's taking from the virus, but it's not directly infecting the virus. It's more like it's infecting the plant and using the virus in along as a side note to that.
Okay, interesting. So just kind of scrolling back up here as I lost my place. What exactly did your PhD? What did you research find?
So there were a couple of things. One was one of the big problems with working with this particular satellite RNA is that it's almost ubiquitous in nature. It actually didn't appear for the first time until I want to say the 70s, and when it appeared, it was really only in the lab. It only existed there. But after they were working with it for a while, it sort of started contaminating more plants, and now there's not a viral isolate that doesn't come with the satellite in tow. So it's very common contaminant. And in order to get rid of it, because I had to do experiments with and without my satellite, I had to develop something called an infectious clone. And what that means is it's a circular piece of DNA that you insert into a bacterial host, and then that bacteria, which is called Agrobacterium tumefaciens, is really good at forcing plants to do what it wants. It will make them express a given protein or RNA sequence, and it will just express it at huge amounts. So I got those circular pieces to work such that they could express my virus, which comes in two pieces. And by doing that, I developed a virus infection that didn't have the satellite as a contaminant. So I had to develop that first. And then when I did that, I was able to test each part of the virus individually with the satellite. And what I found was uh, that certain parts of the virus were necessary for satellite replication, but not everything was necessary. But everything was necessary to both package the satellite and replicate it. So there were quite a few parts to that.
Okay, excellent. So what what harmful effects does the virus have on the plants?
So the virus is called tobacco ring spot virus. It's a very common virus of plants, and we actually don't know the host range, by which I mean it's so broad that we don't know what it doesn't infect. It infects just about everything. Uh, so it's not particularly dangerous to most plants, despite the name. It does infect tobacco, but that's not a bad host for it. The host will just develop these ring spot symptoms on the leaves, which are really cool, but aren't particularly damaging to the plant. The main thing that it does damage to is bean plants, particularly soybean. It causes something called bud blight, which causes the new leaves that the plant sprouts to just shrivel up and die. And it makes it very hard to get a good crop from that. It's not as big of a problem as it used to be, but soybean crops are still badly affected by it. The main
So, what effect does this virus have on the economy? How does this hurt plant growers?
Well, like I said, it used to have a big effect on soybean crops. I don't remember what the specific cost was. I remember it being over $1 billion a year. The current cost is probably well below that. They develop a lot of transgenic soybean, and I think some of that transgenic soybean is actually resistant to it.
Yeah, so I was just about to discuss about genetic modification. There's a lot of genetic modification experiments being done to make plants resistant to various viruses, such as this. How exactly do they create these genetically modified crops?
Well, there's usually a couple of different methods, but the main one is through the same organism that I use for the expression of my virus. So I guess I'll give a slightly longer explanation. The way that it works is that circular piece of DNA is called a plasmid, and the specific kind that this bacteria uses is called a TI plasmid, or a tumor-inducing plasmid. But the main one is through the same organism. And so what you actually see is if you ever go outside and you see trees that seem to have these huge tumors on them, that's the source of it. This organism is actually making those tumors. And the way it's doing that is it's getting the plant to just grow tremendously at the site where it injects this plasmid. So what we'll do is we'll replace the tumor induction gene, the one that causes it to grow out of control, with whatever we want. And what the agrobacterium will do is incorporate that into the plant genome. So we can essentially use a system that's available in nature to get a given gene sequence in there. And then we use what's called a viral promoter. So it's a promoter from a different virus that's very active in plants, and it will activate expression of that given gene, and then that gene will be produced a lot in the plant.
Okay, excellent. And how did you get into microbiology, and why did you choose to do your research on the plant virus?
So I'll start with the first one because it goes way back. My mother introduced me to a lot of different topics when I was young that I became infatuated with, though microbiology was the one that stuck. I first got interested in it when I read Richard Preston's The Hot Zone, which is a story of the early stages of the first, well, not the first, but a sort of 90s era Ebola outbreak. And it was really dangerous and might have come to the United States if not for some efforts to prevent that. There's actually a National Geographic series out for the same book that is really good and worth a watch. But I became really fascinated by how viruses work from that. And it expanded from there to fascination with bacteria and fungi and all microorganisms. And it wasn't a hard sell for me. I liked the idea of working in a lab. I liked the idea of sort of puzzling out a situation and using molecular tools to do it. That just seemed really cutting edge, almost sci-fi, and it captured my interest very easily. As for why I got into plant viruses, I've been interested in viruses ever since I read that book, but I never really explored plant viruses until I arrived at Davis. Davis is UC Davis. That's my college campus, both from undergrad and grad school. Is focused primarily on agriculture, and so in terms of uh, like animals and plants, they're pretty much second to none. And so they have a lot of plant pathology research on campus. And when I was looking into labs, I explored a lot of the plant pathology options and ended up landing on this particular lab. And they just really stewed my interest in it. I got really interested after listening to the PI talk about it for a while and after talking to some of the members of his lab.
All right. And you also did a lot of debates and do debate coaching. And of course, I met you on debate.org, now debateorg.com. So tell us a little bit about your experience with debates.
So I got started in high school. I did debate literally came to my high school in my senior year, and I wish it had come much sooner. But I did enjoy doing it. I did it for that year. I debated with my twin brother, who does not look like me. We're fraternal, and we should not have been a team. I don't think we were a very good team, but we still had a lot of fun doing it. And after I came to college, I saw some note on the back of a chair in front of me during one of my history classes and decided I'd sign up with a friend of mine. And we joined what was a small but thriving debate team and just traveled up and down California to tournaments on the regular. This was parliamentary debate. So it's sort of, if you're not familiar, this is American Parley, so not British Parley. They're 2v2. You get a topic, you get about 15 to 20 minutes to think about it, and then you get to go in and debate it. So on some occasions, you get to research it online. On other occasions, you're not supposed to use the internet, so it gets a little more dicey. But it's a lot of fun, and it's very rapid fire. After I did that for 4 years in undergrad, I was partnered with one of my best friends for most of that time, and that was a really good experience in general. I went to my master's program down in Southern California and ended up there was someone from UC Davis who had gone to a couple of tournaments down in Southern California, and he asked me to judge for a different kind of debate. This was British parliamentary, and it's a little different in a lot of ways. But the main way is there are 4 teams of 2, and they function more like political parties. So you have one side of the debate that's composed of 2 teams, the other side of the debate that's composed of 2 teams, and the sides agree with each other, but they're going to try to win the debate independent of the other team on their side. And it's a really fascinating style of debate. I enjoyed doing it a lot after I came up to grad school. And like you said, I started coaching the UC Davis debate team. That was a lot of fun. It unfortunately didn't last until the end of my time here, but I got to do it for a few years. And it was just great working with fresh sets of students.
Great. And what type of topics do you enjoy debating? What are your favorite topics?
I mean, unsurprisingly, I love scientific topics. Those are the ones that I really get to exercise a little with my professional knowledge. The big problem with scientific topics is your audience requires a lot of explanation, and so it can sometimes be difficult to figure out just how much to put in there. But I still enjoy doing those probably the most. I like political topics in general. I've enjoyed doing very policy based, how are we going to implement a given plan kind of topics. I've also enjoyed straying a little from that and trying some more um, philosophical topics, though I usually like to have them a little more grounded, not vague. I'm not so good at the value style debates of Lincoln Douglas, and I never even tried the incredibly fast talking uh, debates of CETA and policy, but those always seemed like a lot of fun.
Yeah, I've listened to some of those debates on YouTube and was watching them, and I can't even understand what they are saying because they're talking so fast, and they're on one topic, and then once you finally understand what they're saying on that topic, they're on to a completely different topic.
I judged one policy debate, and that was the only one I was willing to do. I barely kept up with them, and there's actually a standard in there that if you have trouble with speed and you're a judge, you can shout clear, and they had to slow down because usually it means they're not clear at all.
Yeah, definitely, and I see that all the time when I'm watching high school debates on YouTube and stuff like that. So I know there is a little bit of a debate going on within the scientific world and among lay people as well as to whether viruses are considered to be alive or are they not alive. So that is the question. What do you think that they should be considered alive or non-living?
It's a difficult question because they do function in many ways like a machine, though the same could be said of a cell in general. Cells are meant to function as machines. That's kind of how they work. It's really the overall amalgamation that forms tissues and bodies that makes it not feel quite so machine-like. Personally, I'd say that viruses are alive. They don't fit the standard definition of life in the way that so many other people like to say it. They don't reproduce on their own. They require a host cell for that. They don't have a membrane structure in every case. Some viruses do. Influenza, for example, has one, but most plant viruses don't have any membranes. They're just capsids. There's a state to be made. There's an argument to be made for why they don't. But from my perspective, they fit way too many of the criteria, and beyond that, they seem they. I my impression of it is that we're pretty lenient when it comes to that for bacteria because there are bacteria that also require their host for reproduction. I don't see why we can't be any lenient at all for viruses.
Right. And of course, the more you study something in nature, the more complicated it seems. And the more you try to categorize things, the more you see that things just don't necessarily fit the neat definitions that you want them to fit.
Yeah, I think a lot of our definitions for life, especially, are based in what looks like us, what's similar to humans, and that is, in my opinion, out of date and a little anthropocentric.
Absolutely. So, what is your next step? What do you hope to accomplish with your PhD?
Well, I mean, I don't need to tell you this is probably the worst possible time to graduate from a PhD program, from any program. I've had the worst timing. Back in 2008, I graduated from my undergrad just in time to get into that recession. Back in 2011, I graduated from my master's program just in time to have Genentech fire half of its R&D staff, which meant they flooded the market with people. This time it's difficult as well. I'm having trouble securing something. But I mean, I'm looking into public health positions. I'm looking into biotech positions. I'm looking into government research lab positions. For the most part, I'd be interested in an industry career. I like the prospect of applying my science more. A lot of my PhD was very basic science, establishing that something is going on, rather than trying to apply some basic science that we'd already understood to some larger issue. I like the application side more. I feel like it suits me better. I see the bigger picture more easily than a lot of my contemporaries, and I feel like it's where I belong.
Great. Thank you so much for coming on. And is there anything else you would like to say, or anything you would like to point out about your research?
I guess one thing I can talk about is just what it takes to sort of be in that startup phase for a biotech company, because that was very interesting and very new for me. I didn't really get to experience. I don't know in any other environment how I would have gotten to experience that. But the basic idea was we had this initial research that I'd done at UC Davis, and we ran out of money through a grant through the university, and then we were trying to find money for it and decided that spinning off a company was a good idea. So we actually recruited a postdoc from my lab to take on the role of CEO, and he and I commuted out to San Francisco from Davis 5 days a week by train. So we were up at like 5:30 every morning, and up and out at 5:30 every morning and back at 7:30 every night. It was a ridiculous schedule, but it was still incredible. The idea was we were actually at a incubator slash accelerator in San Francisco doing this research. That's the reason we were trucking out every day. And by the way, he had an infant child during that time. I don't know how he survived that. I don't know how his wife didn't kill him. That must have been insane. But it was a really amazing experience because we were building from what was very basic science at the time that we barely established into an actual product. And so we were working with people who were familiar with the early stages of biotech companies. They were helping us build a lot of the basic tech. We were making prototypes on the fly to make it work. I'd never prototyped before at all, and I was like gluing sticks together to make something work. Like we were literally sticking magnets on the end of sticks because the idea was we had a specific protein and we wanted it bound to the magnet so we could move it from tube to tube, and we were gluing these magnets onto the ends of sticks, taping them up so that it wouldn't be contaminated by the solution, and then just passing it from tube to tube. And it was ridiculously a shoestring budget kind of situation where we were just rolling as fast as we could to get what we needed to do done. We were actually 3D printing some of our stuff at their site because they had a 3D printer on site, which was really nice. But even though the company didn't take off or do anything, it was a really valuable experience. It's probably the most valuable experience in my PhD, despite the thesis and everything. I feel like this is the one that's going to stick with me the longest, and it's going to be the thing that I remember the most and appreciate the most. So yeah, that I would say that's the main part of my grad experience that we hadn't already covered. I don't really have anything else.
Great. Well, thank you so much for coming on and talking about your research with me. And once again, thank you so much for being a part of our community. And I'm definitely looking forward to seeing what you discover later on in the future with your research.
Thank you. I appreciate that.
All right. We are signing off.
Bye.